here I am writing, they are from after exam 2. Correct? After exam 2, you will have 180 points. Again, if you are not understanding and if I am saying something, I might be thinking something but saying something different. That happens. Okay? Please ask me. Yes?
You said it was 150 points first, so is it actually 200 points?
Oh, thanks a lot. I didn't change. Hold on, sorry. So what is it coming? 10% of 150 is how many?
15.
Sorry. Thanks a lot. That's a very important thing. 15 points, right? So 15 points. Oh no, 200 points. Is it?
No.
Yeah, final exam is 200 points, right?
Bet you lowered it.
In your exam 1, how many points? 150? So my mistake. That's correct. Your final exam is out of 200 points. Okay? So a final exam is out of 200 points. Sorry, my mistake. Alright. So everything is fine here. So from 200 points, 20 points are allocated for exam 1 and exam 2 content. Alright? And 180 points would be from after exam 2. Now what is after exam 2? From lipid. Okay? From lipid, everything after that. So lipid, DNA, RNA, all the DNA bases, nucleic acid, then nucleotide, all those you go, and then you go to protein synthesis, DNA replication. Yes?
Are you going to post like exam 1 and 2, like the answer sheet, so we can go over those too, or no?
They are from the activity. No.
Okay.
Also, from exam, after DNA, RNA, protein synthesis, you are going to get what? You are going to get drug discovery. Okay? After drug discovery, you have virus. So not too many things. Okay? Go through all the lecture notes and all the activities that I have done. Now remember, activity doesn't contain everything. So you also need to do the lecture note. Okay? Everyone got it?
Mhm.
So lecture note, activity, and there might be like 5, 10, 20 points from the book, from the homework. Very little point, 5 to 15 points will be from the book. Okay? From the homework. Alright, so this is our intermolecular force. Again, these are just hydrogen bonding. Then we have dipole-dipole interactions. We have hydrophobic, hydrophilic interactions. Mainly, I want to know the interactions, whether you know the interactions or not. In the biological molecules, what kind of intermolecular forces are there? You have to identify them. Okay? Then we have draw the peptides. Peptides, you know, glycine, then alanine. If you have glycine, alanine, and if I say draw the peptide bond AGV, can you draw it? Alright? And then we are going to talk about 3D structure, primary structure, secondary structure, tertiary structure, and quaternary structure of protein. What are they? Primary deals with sequence. Secondary deals with what? Helical structure, beta sheets. Tertiary means what? It is the overall 3D structure of a single unit of the protein. Quaternary means you have multimer, like a monomer, dimer, trimer. These are multimers. They can also, their protein can also be like that. Then protein denaturization, all of you know that if you raise the temperature, protein denature, like an egg, you heat it, it becomes hard, right? So, and then acid-base properties of hemoglobin and their binding. So there was a question I asked you, and it is also in your activity, how hemoglobin control takes the oxygen from our lung to the whole body, right? What is affecting? Why the hydrogen and carbon dioxide are coming out, right? So these questions are mainly in the activities, okay? Acid-base property, predict protein structure from bioinformatics. That's also actually relevant to your drug discovery. Remember the Swiss model, we discussed Swiss model, homology modeling, AlphaFold, these are there. Be able to explain different ion channels and transporters. Understand how rate change. Be able to determine reaction order. So these are from enzyme. So I'm just asking what is the order. Vmax. Remember Vmax, the picture of the V. Vmax is what is the Vmax, guys? Maximum velocity of a enzyme. Can you draw that structure? If I give you a structure like this, where you have the substrate concentration, and this is the V initial, like that, right? This is the Vmax here, Vmax is here, right? And if I give you this thing, can you tell me what is the Km? If I give you a picture like that, can you tell me what is the Km? Let's say this one is 1, this is 2, this is 3, this is 4, this is 5. Can you tell me what is the Km here? You say, okay, this is the 50%. The corresponding one is here, right? So it's going to be around 2.5, 2.2. So this is our Km. Km, is it like that? I forgot the, yeah. Anyways, Km. Okay? Michaelis constant, right? Okay. Be able to use and understand reaction energy diagram. If I give you a diagram, can you say whether it is exothermic or endothermic? Whether can you say what is the activation energy? Can you say whether it is going to be a spontaneous or non-spontaneous? Can you say what is the activation energy? Remember, this is what we discussed. Now you tell me, what is the activation energy here? This one, right? How many steps are there? 1 or 2? 2. What is the, remember, these are actually within 80%. I would actually change those things. These goes everywhere. These are not only within 20%. These are also going to drug discovery. Same like bioinformatics, we saw this thing. These are actually going everywhere. That's why I have to put this here, or I can put it in the drug discovery, because they are going, whenever we discuss about biochemistry, they are always going to be coming. Okay? Okay, so if I give you this barrier is 200 kilocalories. This one is 50 kilocalories. What is the activation energy? What is the value of the activation energy? 150, correct? Take the difference from here to here, right? What is the delta G? Let's say this is where is so it is 10 kilocals. This is 50. What is delta G? Negative 40. Good. Negative 40. So these are basic stuff we need to know. Okay? All right. Okay. Be able to identify reaction intermediate and catalyst. Reaction intermediate is which one here? Where is the reaction intermediate? Can anybody tell me? This one is the intermediate. Okay? Catalyst is not here, but if I use the catalyst, what is gonna happen? Activation energy is gonna lower, right? So it might be like that. One step, or if it is two step, it's gonna be like this. Basically, it's decreasing the activation energy. Okay? All right. How enzyme affects the reaction and activation barrier? Enzyme lowers the activation barrier, correct? Okay. I'm going to spend more time on this part, but I'm just quickly going over this. Okay, so what is enzyme? What is cofactor, coenzyme? Activation, inactivation. Just definition. Competitive and non-competitive inhibition. Competitive and non-competitive inhibition, they are also going to come in the drug discovery. We discussed that. So these are going to be competitive inhibition is also in the drug discovery thing. Write different names of the enzymes and 6 major groups. Can you identify reactions according to the classes of enzyme, the name? Can you give me a name of a enzyme? Oxidoreductase, remember? Lyase. So these are the ones. Okay? All right. Now let's focus on this one, and I'm going to be spending more time on this. This is very important because you will get most of your questions from here. Lipids. No lipids. What are the lipids, guys? Huh? Lipids are molecules that contains hydrophobic head group, hydrophobic tail group, and hydrophilic head group. Okay? That means hydrophobic and hydrophilic part is there. Okay, different classes of lipids. Can you tell me?
Triglycerides, fatty acids.
Huh?
Triglycerides, fatty acids.
Yeah, triglyceride fatty acids. And then can you tell? Okay, so these are the ones. Fatty acid, triglycerides. So you need to know the structure, at least the structure. What are the components there? Remember, go over the lecture notes. Triglyceride. What are there? What are the things there? 3 glycerols. And then you have the fatty acid coming out, right? Sphingolipid. What is there? Sphingolipids, glycolipids, cholesterol. So look at the lecture note and find the structural components that are there. Ceramide, sphingolipid, ceramide is there, and then there are the fatty acids connected with a amide group. Okay? Be able to differentiate saturated and unsaturated lipids. Everyone should know that, right? There is a key. Which one has higher boiling point? Saturated or non-saturated? Unsaturated? Huh? Which one is the saturated one? Huh? Has no king. That means what? Do they have double bond or single bond?
Single bond.
That's the important part. Then you can go on and say other things, right? Okay. If you increase the number of carbon atoms, what happens to the boiling point?
Increases.
Why?
Higher molecular weight, more kinks in
It is more. If you have more carbons, that means you are increasing the molecular weight. If you increase the molecular weight, dispersion force increases, right? Then comes if you have more double bonds. what happens?
Lower boiling point.
Then boiling point decreases. Why? You say it has more double bonds, so it has more kin.
Yeah,
They cannot
more together.
have more intermolecular interaction within them, right? Okay, explain different lipoprotein and their function. All of you know that there are 4 of them, right? You have to explain it. How the lipids or fatty acids are transported from intestine to the liver, from different tissue to the liver. No disease-associated lipids, blood group. Blood group. What are the groups that are found in A and B?
Glycosyl transferase.
Eh? And what enzyme is making it? There are 2 different types of sugar, galactose, I think. And another one is,
Acetylsoluble.
yeah, another one is something like that. It is another, but O doesn't have that. O doesn't have those.
No.
Let me open that. What was the name? Okay. Activity, no, lecture, right? Oh, yeah, here you go. So everything is here, you see? You have to know what are the fatty acids, unsaturated, then melting point decrease with degree of unsaturation. That means with the number of double bond increase. Okay? That's called degree of unsaturation. That means you have more double bond. Here you have more double bond, you see? That's why boiling point is lower compared to here. Okay? And then these are triglyceride. Triglyceride, you see, these are the glycerol. 3 fatty acid is connected with the glycerol. And then solubility. Solubility, we discussed about that. So which one is bad? If unsaturated is bad, right? So you have to be careful about that, right? And then phospholipids. What are the phospholipids? Yeah? Phospholipids. What are the differences in phospholipid? What are the different components in phospholipid? It is a head group, phosphate, glycerol, and the fatty acid. You have to say that. I really don't need too much structure, but I need those things. I need to know what are the components there. Okay. See, sphingolipid in sphingosine with fatty acid. While multiple cirrhosis is happening. Yeah, so this is the one. So this is N-acetylgalactosamine for A blood group. You see, I remember the N, but this name is very big. Acetylgalactosamine. But for the B group blood group, you only have the galactose group. Okay? O doesn't have anything. Now, what enzyme is making this? It is called this one. What is it? Glycosyl transferase enzymes are doing that. They are making this too. So for some people like me, I have O group. I don't have. I might have glycosyl transferase, but it is not active. It is not making these 2 ingredients in my blood. Okay? Alright, so these are the things, yeah, so this is vitamin D. How does vitamin D synthesize? And then you explain how it is doing it. Okay? What is the purpose of vitamin D? Then these are the lipoproteins that are used, that are involved in transporting the lipid, fatty acid in our body. Okay? we need to know the process. Intestine, then it goes to the liver, to the tissue. Then from the tissue it again comes back. HDL and LDL, very important. All right. Oh yeah, so we also discussed about membrane transport, passive, simple diffusion and facilitated diffusion, lipid bilayer. These are all in lipid chapter. These are all in lipid chapter, lipid. So we first finished the lipid and then we went to chapter, the next slide is lecture 8. So in lecture 8, we discussed about these things. So transfer. These are the ones. Able to define and distinguish DNA, RNA, nucleic acid, nucleotide, DNA, and ribonucleones. So these are the structural component of DNA and RNA, right? So we need to identify and define them. We have to say, okay, this is adenine, this is guanine, this is cytosine. Okay, and then the primary, secondary, and tertiary structure of DNA. For example, DNA is a helical structure, but it has 2 strands, right? Cytosine goes with guanine, right? Thymine goes with what? Thymine goes with adenine, right? So these are the things, DNA denature. So basically what I have here is in chapter 8, we already discussed in chapter 8, lecture 8, sorry, not chapter 8. So these are the ones. So you have to identify the base, okay? This is the base, this is the deoxyribose, and this is the fatty acid, this is the phosphate group. All right, so this is for RNA. And then these are the cytosine, thymine, uracil, adenine, and guanine. All right? And this is another one, this is the ATP. ATP is also here, so you see ATP means only one, AMP means only one phosphate group. Then D means tri. Okay, there was a question in your quiz, last quiz. Remember guys, the last quiz was on DNA, RNA, and nucleic acid. I asked a question related to this one. ATP hydrolysis means reacts with water forming this. And it is also releasing energy, right? And I said, draw the energy diagram. I saw many of you were not writing this answer. So, what is that? So this is just like a reaction, right? Just like what I wrote here. You are going from reactant and through a activation energy and it is going to a product. 7.3 kilocalories of joules of energy is released. Release means it's going out. That means you are removing it, right? That's why it is on the right side. 7.3 is on the right side because it is coming out as a product. That means it is coming out as a product. Reactant and product, that's product, okay? So that's why the energy of the system is getting lower. Energy of the system is this plus this. This one plus this one is the product plus 7.3 kilocalories that we have gives you this energy. Then you go here. Okay guys? So that's why since it is coming out, this energy is released, and to say it is released spontaneously it is releasing, we actually, to show it is a spontaneous, we put a negative sign. Are you guys? So it is exothermic because it is going out of the reaction. So if I ask a question like that, can you draw a picture like that? And say that this is the reaction energy, that is what is this 7.3 kilocalories of kilocalories mean. Okay, and then we are talking about the DNA structure. You see DNA. Again, not too many lectures, like only lecture 7, 8, 9, and 10, right? For the exam. Mainly, 90% of the questions are from there. All right. So you see here you have a nucleotide that is connected with another one. It is by 5-3-5-3. It starts from 5, finishes at 3 for one of the DNA bases, the DNA strand. For the other one, again, it goes from phosphate, sugar, phosphate, sugar. Base is just adding, right? This is the base with the sugar. All right, so this is what we learned, difference of RNA and DNA. All these things are important. I don't know how to ignore any of those things. You have to read everything in the lecture. If you look at this, not too many things are there, okay? So what is the DNA denature? Denature means you are making from double strand to single strand, that's all, right? How can you do that? By raising temperature, changing the pH, by adding solvent. All right, so what is this one? Melting temperature. Melting temperature is lower if the DNA is unstable. That means you can change the DNA. If you can make a DNA denature at a lower temperature if its melting point is lower. You see, this one is 80, this one is 85 or 82, this one is 85. So if you raise the temperature for this one, since the temperature is PM, which is the melting temperature is low, you can separate them. Okay, then comes this. What is the DNA superstructure? What is it made of? Histone. Histone the DNA is surrounding the histone. And the histone is are positively charged. Lysine, arginine. Remember, lysine is not leucine. Lysine. Okay? So leucine and leucine are different than lysine. Okay? Then DNA is negatively charged. So that's why they want to come close to each other and they form the nucleosome and they make the chromatin and that's how we get the chromosome. Right? So, and then we are going to go to replication of DNA. What are the things involved? If I ask you a question, again, there will be some descriptive question. I'm going to ask, what are the things that are involved in DNA replication? Explain. Okay? You have to explain it. If you just say, okay, this is either 1 or 2 lines, you are not going to get points. Okay, and then, so this is the replication of DNA, and from this replication, we get new DNA. All the processes are here. And then, and again, I'm gonna check the plagiarism or anything. Don't just copy and paste because it's an online thing, right? Don't paste, copy and paste. You have to write it in your own word. Okay. Then protein synthesis. Protein synthesis, okay? Yeah?
You said it was online?
Yeah.
Okay.
You just came? Yeah, I explained everything. If you just came, I'm just letting you know, we are gonna have the exam virtually, like with Blackboard. Okay? I'm gonna send a more detailed information. I'm gonna let you know where to go on what day. Please follow the instruction and then you will be able to do it. Okay? everything will be on the instructions. The questions will be multiple choice questions, fill in the blank, mix and match, and descriptive questions. All right? All right. Yeah, here you go. Okay, then comes, now I know for most of you, you know the lipid and the nucleic acid and DNA, RNA. The problem might come for the last two chapters. Well, I don't know, because we haven't, I'm not sure if you guys are studying or not. I'm not quite sure. So, what I'm doing is, I already posted the activities, right? Are you guys doing those? Please do the activities. All right. So we discussed about different ways of developing therapeutic drugs. One of the ways is to do something called docking calculation. Docking, whether a molecule binds with a protein or not. That's one of the important things that we always want to do. Let's say you are developing Tylenol. You take Tylenol somewhere, right? All of you take it. If you have a headache, you take Tylenol, right?
Mm-hmm.
The Tylenol is a small molecule. Okay, it has thousands of a small molecule makes that Tylenol, that small thing. It's a small molecule. It has a structure, organic molecule. That molecule goes inside our body in a certain place where it binds in a protein. Then it triggers a response. So we want to see if a molecule binds with a target protein. So that's why we do the docking. We use thousands and thousands of molecules and say, oh, okay, let's see if those molecules bind with the target molecule or not, protein or not. Okay? That's what is called docking calculations. And docking calculation, so what is the docking calculation doing? If I ask you, how do you develop a therapeutic drug that binds to a particular protein? What are you going to be asking? Saying. You say, we are going to be doing docking calculations to see, or docking, we will be doing docking to see if the molecule binds to the protein or not. Okay? Docking can be done. You don't stop there, you have to explain more. You say docking can be done in two ways. One is called the structure based, another one is called affinity based or electrostatic. Structure means what? Whether a protein is there and it has a small hole, whether the molecule is going to go there or not. Okay? But affinity means whether there are hydrophobic group, and here you also have hydrophobic group. We know hydrophobic like hydrophobic, right? So there are intermolecular interactions involved. Having a small pocket to bind is not enough, because that might be hydrophilic. There might be hydrophilic groups, amino acids. They may not like hydrophobic molecules. You understand what I'm saying? Hydrophobic molecule doesn't like hydrophilic molecule. Like diazoxide. We discussed that before, right? So similarly, the drug molecules also work like that. Hydrophilic likes hydrophilic. Polar likes polar. Non-polar likes non-polar. Okay? So we do that through two ways. One is shape. Shape means the pocket, to identify if there is a pocket or not for the drug to bind. And then another one is called affinity based, whether there is intermolecular force that are contributing towards the binding of the ligand. Okay, so we have done up to this one: DNA duplication, peptide protein synthesis, drug discovery process, explain strategies for drug discovery. You say, okay, if we are discovering a small molecule, it has to bind with the target protein based on two ways: shape, another one is affinity based. Okay, shape as well as affinity has to favor. Another one is protein based. Protein interaction has to be there. So sometimes we develop therapeutic drugs that are protein based, not a small molecule. Okay? Protein binds with another protein, target protein. So if you have a protein, let's say this is our protein, that is the SARS-CoV-2 virus protein. If you are developing another protein, let's say this is another protein, whether it binds or not, that's important. If it binds, that means it can be used as a therapeutic drug. Now, how do you improve? How do you make a protein to bind with the target protein? This is the target protein, target. How do you do that? By knowing the amino acids that are here, if they like these amino acids in the protein. Okay? If they are hydrophobic, they are going to like it. If they are hydrophilic, they are going to like it. And then, in drug discovery, we want to make sure they bind more. So what do? The amino acids that are involved in binding are called hotspot residues. Hot spots. These hotspot residues bind with the residues in the target protein. Okay? And hotspot residues are important. If we don't have the hotspot residues, they are not going to bind. We can improve the binding by mutating the residues, hotspot residues. Let me write it like that. By increasing the binding, let's say arginine is here, it likes lysine. They bind. That's the arginine and lysine are hotspot residues. Now you say, I want to improve the binding. Let's say you do a mutagenesis, that means you change the arginine to 19 other amino acids. Now you say there is a glutamic acid, or let's say there is some other amino acid, let's say histidine. You see the binding is even more now. That means you are now designing the protein to make it improved drug. You understand what I'm saying? So this is called hotspot residues can be used to make the drug more potent so that they can bind with the target protein. When you take it, it's going to look for a protein, and then all of a sudden when it sees it, it's going to say, oh, I now want to bind it. You understand what I'm saying? Bind means attach to this. Okay. So these are drug discovery, experiment, potential energy function. Another one is potential energy function. We discussed that. What are those things? These are potential energy functions. So a protein system or a biological system can be explained in intermolecular interaction and intramolecular interaction, inter and intra. Okay? Intermolecular interactions, intramolecular, intra means within, can be explained with this energy. So this is stretching, this is angle bending, and this is the dihedral torsion. And these are the non-bond interactions. So what is the potential energy? This is the potential energy. Describe molecular systems such as protein, DNA. More potential energy function. Explain potential energy function. I just explained it. Read that. How competition can help discover therapeutic drugs? You can do it quickly, efficiently, and you can see whether they are going to bind with the protein or not within a second or a minute or a second. Okay. Okay. Define molecular docking. I already explained. Free energy diagram depicting energy diagram of a dissociation of a protein. Formation of dissociation of a protein. Can you draw a dissociation energy of a protein? So this is the one. So if you have a protein system like this, protein binding with a ligand, forming protein ligand. So what you are going to have, protein binding with a protein and ligand, they are separate from each other. If they like, they are going to bind, right? If they like, they are going to bind, their energy is going to decrease. Always remember, if a molecular system is after reaction or after binding, if they become more stable, their energy decreases. Okay? Just like what we saw here. This product is more stable than the reactant, so its product energy is lower than the reactant energy. Same here, protein and ligand energies are higher, but when you add them together, their energy decreases. Okay? All right, so this is what I was asking. Free energy diagram. Explain mutagenesis and how it can be used to increase binding affinity of a peptide or protein. Mutagenesis is this one. I said if you have arginine, then you change it to histidine and all other 19 amino acids, and if they increase the binding, that means they are gonna, that process is called mutagenesis. You are changing one amino acid by 19 other amino acids, one after one at a time, like one at a time. So that means you are looking at, after changing the mutation, after doing the mutation, whether they bind or not, whether they are increasing the binding or not. You are checking that. How it can be used to increase binding? So after you just check, you just calculate the binding affinity. Okay, how change in define molecular dynamics and how molecular dynamics can provide change in free energy. So molecular dynamics means you have a protein system, you just allow it to move by using Newton's law of motion. Newton's law of motion means F equals ma. F ma, I have it here, let me explain it. So this is the F equals ma. Force equals mass times acceleration. So this I means a single atom. So all the atoms are moving. We have thousands and thousands of atoms in a protein, right? And it is also dissolved in water. So we are calculating the force of the system, total force, if we know the force for single atom. And we can calculate it from the acceleration times mass. Okay? So over time, because acceleration is dependent on the time, because it's the r over t, position, change of position with respect to time. That's called the acceleration. So if we know how it is changing, we can look. This is what is happening. Look, every femtosecond, you see the molecule is protein is moving. You see, protein is moving. So by looking at the protein, how the protein is moving with the water, these are the waters, these are the ions, we can mimic the protein's motion that is happening in our body. That's called molecular dynamics. Okay? So this is the molecular dynamics. Some free energy method related to molecular dynamics, such as docking, is one of the free energy calculation method, whether they bind or not. Another one is called MMGBSA, which is, I explained it in detail in the class activity. Please look at those answer sheet that I gave. So basically, MMGBSA is again, you are calculating whether they bind or not. A ligand is binding with a protein, and you are calculating the intermolecular interactions, internal energy, electrostatic energy, van der Waals energy, and all those things energy, and the total will give you the MMGBSA energy, which is I'm showing here. So here you see total energy. If you add this 4, it is going to give you the final free energy of binding. That's another method called MMGBSA. Okay, so we are almost there. So then comes virus. Define virus. What is it? Viral particles. Example of the various viruses. 4 types. Remember. Know how different viral fusion occurs. Pathways. Remember the path. Viral pathways. Viral life cycles. And then how HIV and SARS-CoV-2 attach to the cell. SARS-CoV infect and replicate virus. How do they do it? So these are the things. IC50, what is IC50? Half inhibitory concentration. So please make sure you know all these things. Just follow whatever is there, and then if you don't understand, you can come tomorrow to my office hours or on Friday. But follow these steps. Whatever I'm showing in the topic, make sure you know everything. Then you will do very well in the exam. Okay? And last but not the least, you saw now it's the open book basically. So you don't need the reference sheet. I posted the reference sheet, right? With the periodic table. You can print it out and okay? Yeah.
You sent an email to me to email you about extra credit. That's not the.
That what?
You just sent an email to me. On the website. You mean like
You actually separated methods and shit. Yeah, I'm sure that